Hello students, we will be discussing about the importance of Anthoceros, which is a very important genus, no doubt. We will be discussing its important characters. The topics covered are similarities between Chlorophyce and Anthoceros, 2. Anthoceros Sporophyte, 3. Evolutionary significance of Anthoceros. Now answer. Anthoceros and Chlorophyce share the following main similarities. What are these? These are, number 1, chloroplast in both contain chlorophyll A and chlorophyll B as their photosynthetic pigments. 2. Chloroplast is surrounded by two membranes. 3. Starch in both is the primary food reserve. 4th, cell walls in both are cellulose based. Now these 4 characters are common to higher plants also. Chlorophyce, Anthoceros in all higher plants. Now the other 3 characters are common to Chlorophyce and Anthoceros only. Now what are these? 1. Some Chlorophyce and Anthoceros have pyrenoids within their chloroplasts. Now I have said some Chlorophyce. What are these? These are, at the bottom you can see with the star mark, Chlamydomonas, Volvox, Eleutheryx and Spirogyra. Now the Spirogyra, Eleutheryx, these are filamentous and is included in the Zygnematoceae. Now the next point is flagella in both is of whiplash type. Now the flagella is common in Chlamydomonas and other Chlorophyce. In Anthoceros, where is that flagella? Flagella is in the antherozoid, and in both it is of whiplash type. And the last point is plant body in both is of thalloid type. Now what do we mean by thalloid type? That means that the plant body, the tissue of the plant body is not differentiated. It's just a thallus structure. Now these are the key similarities between Chlorophyce and Anthoceros. In a sense, these similarities highlight the evolutionary link between aquatic green algae like charophytes, Anthoceros and land plants. Now within charophytes, special mention may be made to Chaetophorales and Zygomatophyceae. Now, in the bottom you can see an asterisk. It is written that previously Anthoceros was linked to Chaetophorales, example Colocyti. This is done on the morphological basis like heterotropous plant body. But later genetical studies and ultrastructure revealed closer relationship with Zygomatophyceae, like the presence of pyrenoids, etc. Though these two are morphologically different. So it seems safer to refer the link to some charophyte algae. Our next topic is Anthoceros Sporophyte. On one side is the diagram of Anthoceros. You can see that the long, slender sporophyte is arising from the gametophytic thallus, which is at the base. The base of the sporophyte is partly covered by the involucre. This involucre is a gametophytic structure. So, Anthoceros Sporophyte is a long, slender, green, horn-like structure firmly attached to the gametophytic thallus at the base and surrounded at the base by involucre, a gametophytic tissue. Because of the horn-like structure of the gametophyte, members of the group of Anthoceros are commonly called hornworts. Before we go to the description of the interior of sporophyte, let us discuss the importance of Anthoceros sporophyte. The sporophyte of Anthoceros is unique among bryophytes due to the following characters which have evolutionary significance for being the forerunners of mosses and pteridophytes. Now, what are these characters? Number one is the prolonged growth due to the presence of intercalary meristeme, and this is the first time of the presence of intercalary meristeme in the case of bryophytes. Secondly, previously all the bryophytes like Rixia up to Paleoporella do not have chloroplast, and hence the sporophytes are unable to photosynthesize and are completely dependent on the gametophyte. But here, for the first time in bryophyte, the sporophyte, there is the photosynthetic capability that is due to the presence of chloroplast, and they are partially independent of the gametophyte. So the second point is photosynthetic capability, for which it remains partially independent of the gametophyte. Next is the internal structure of the Anthoceros Sporophyte. At one side you will find a partial diagrammatic representation of the longitudinal section of Anthoceros Sporophyte to show you the location of the different tissues. See here the no cells of the tissues except for the sporogenous tissue is drawn. Now the sporophyte, as you can see, consists from base upward. Number one is the foot at the base, you can see in the figure. Now it is a bulbous parenchymatous structure at the bottom. The foot in some species possess haustorial process which penetrate deep into the tissues of the gametophyte. For anchorage of the sporophyte to the gametophyte and also absorbing nutrients and water from the gametophyte. Now, in all the species there may not be any haustorial process, but the function of the foot is the same, that is, they anchor the sporophyte to the gametophytic tissue and also absorb nutrients and water from the gametophyte. Next, the important point is there is no setter. Okay, mark this, there is no setter. Then the structure is the intercalary meristematic zone. You can see in the figure just above the foot is the meristematic zone. Now the location located between the foot and the capsule. Capsule is the upper portion, you can see it. This basal meristeme continuously add new cells to the sporophyte, enabling the continuous elongation of the capsule. Next is the capsule. It is above the meristematic zone, as we discussed earlier. Capsule or sporangium. Why? Because the capsule is the structure which produces spores. Now, first of all, we have already described earlier that it is a cylindrical and long structure. Now, the internal structure within the capsule, we describe it chronologically from the center to the periphery, as that is seen in the figure by the side. First of all, the central tissue is the columella. This tissue consists of elongated cells arranged in 16 vertical rows. its function, number one, is provides mechanical support, and number two, which is the more important, that is allows conduction of water and minerals. This function is the first of its type in Bryophyta. Now, surrounding the surrounding the columella is the archosporium, that is the sporogenous tissue. This tissue is single layered at the base but broadens upward. It gradually differentiates into first spore mother cells, which is a diploid structure, diploid cell, 2N. This undergoes meiosis to form haploid spores, 4 in number, that is spore tetrads, and then 2 free spores. Now in meiosis we have 2 divisions. First is the reduction division and second is the equational division. So in the first division, spore mother cells, from the spore mother cells we have 2 N cells, that is reduction division, and from 2 N cells we get another division, equational division. Finally there is 4 N cells, that is 4 tetrads, and after this the spore tetrads gradually separates to form free spores. In the last slide we have seen that from the archosporial cells, that is diploid cells, 2 types of cells are generated. One is the fertile spore mother cell, 2N, and another type of cell, that is the sterile elater mother cell, which is also 2N, that is diploid. We have already discussed about the spore mother cell, which divides mitotically and finally give rise to 4 spores. Now we come to the sterile elater mother cells. These cells are also diploid, 2N, and these cells divide mitotically. So that is the equational division to form pseudoelaters, which also are 2N. Now these pseudoelaters lack the typical spiral thickening of the true elaters. That is the only difference. The function, just like elaters, pseudoelaters aid in spore dispersal. Now the next layer, that is the capsule wall or jacket. Now surrounding the archosporium is the capsule wall or jacket. It is multi-layered, having 4 to 6 layers consisting of the outermost layer, the epidermis. Epidermis is cutinized, vertical cells, and interrupted by stomata. Now interior to this is the inner chlorenchyma. These are parenchymatous tissue containing chloroplast. Therefore, it is the photosynthetic tissue supplying additional nourishment to the sporophyte, supplying nutrition and making it partially independent. Now we come to the detailed diagrams. This is Anthoceros Sporophyte, full sporophyte, transverse and longitudinal sections. On the right side of the page and on your left side, there are serial transverse sections taken from the lower, middle, upper middle and upper portion of the sporophyte. In the 3 lower ones, cells of the jacket is not drawn, and the upper one shows all the cellular types. On the left side is the vertical section showing top to bottom cut from the middle. The longitudinal section shows all the cell types and particularly the development of the archosporium from the bottom to the top at a glance. Now in the longitudinal section, from the bottom we find the foot, then the meristematic zone. The thallus, where the foot is anchored in the gametophytic tissue, And involucre also is a gametophytic tissue and not part of the sporophyte. And these facts should be mentioned. So, the thallus where the food is anchored and the involucre are gametophytic tissue and not a part of the sporophyte. This should be mentioned. The meristematic zone is followed upwards in the longitudinal section defined by three tissue types. One, the outermost, the jacket cutinized epidermis, interrupted by stomata. This epidermis has vertical cells, beneath which is the parenchymatous cells of the jacket with chloroplast. Chloroplast could not be shown here because this is out of the scope and could not be drawn. But the different shape of the epidermal cells and the rest of the parenchymatous cells of the jacket can be seen if you can zoom a little. In the center is the columella, made up of elongated cells. The development of archegonium from the base to the top is shown in the figure as well as is represented schematically. Now in the scheme you can find at the bottom is the archosporium, just containing cells, diploid cells. Now this archosporial cells divide into two types of cell, spore mother cells, diploid, and elater mother cells. This is also diploid. Now the spore mother cell undergoes meiosis to form four cells haploid it in the form of a spore tetrad. And the elater mother cell divides mitotically to form pseudoelaters, which are diploid. The spore tetrads, which are aggregated spores, 4 in number, then becomes free and are just round spores, haploid spores. Similarly, the elaters, pseudoelaters, are free pseudoelaters, also they are diploid. So coming back to the figure, the transverse sections, first near the bottom, is the smallest, showing a row of archosporial cells around the columella. Surrounding the archosporial cells is the jacket with epidermis. Jacket cells are not drawn here. Note the archosporial cell is one layer. Archosporial layer is one celled only. Transverse section near the middle shows archosporium wider, showing the development of spore mother cell and elater mother cell. Correspondingly, the longitudinal section also shows the spore mother cell, which is roundish and bigger, while the elater mother cells are smaller, elongated. Still above, in the section near the top, we find sporogenous area is wider with spore tetrads and elongated multicellular pseudoelaters. The same thing is seen in the longitudinal section. In that position, the uppermost section, both in the transverse and longitudinal section, shows the cells of the jacket also. Here the sporogenous area is wider, showing free round spores and long pseudoelaters. This is again the longitudinal section of Anthoceros, where the lower portion is not seen clearly. However, the rest is bigger, hence clearer. You can see here the epidermis, jacket, the columella, and archosporial area in different stages of development. The spore mother cell and elater mother cell is clearly seen. Still higher up, the spore tetrads and elaters, and lastly at the top, free spores and multicellular elongated pseudo-elaters. Now, this is the last diagram. The same longitudinal section of Anthoceros Sporophyte, but it is partially diagrammatic because all the cells are not drawn. The cell types in different regions are shown clearly. Archosporal development is also shown fully in one side. This is just to show that in the examination hall, full sections is difficult to draw within a limited time. So this type of diagram can be drawn more easily. We will be now discussing a very important topic, that is evolutionary significance of Anthoceros. We have discussed a little earlier, but now we are going for a more detailed discussion. A little introduction before the answer. Life first originated in water. Over the years, plant life migrated to land and gradually evolved into a highly evolved land plant. In the evolutionary scale, we find, starting from the early or primitive to the more evolved, first algae, then bryophytes, then pteridophyta, gymnosperms, and lastly angiosperms. Now, what are the main difference? Algae is aquatic, bryophytes are amphibian, rest are land plants. That is, from aquatic to land plants is the evolution. Bryophytes are gametophytic plant body, while the land plants have autotrophic sporophytic plant body independent of the gametophyte and have vascular system for conduction of water and nutrients from the soil. So these two properties, gametophytic plant body and sporophytic plant body, and secondly the independence of sporophytic plant body, are the major features of advancement. Now, Anthoceros represents an early lineage of land plants. Answer. Anthoceros are considered to be a synthetic group because they have both an algal lineage as well as they possess characters that may be considered as forerunners to pteridophytes. Now, first of all, the characters of algal lineage. Now, this I have already discussed. If we turn to page 2, I am just reading it out because this is included in the answer. Now, the points: some chlorophysi and Anthoceros have pyrenoids within their chloroplast. I have mentioned the name of the chlorophysi in page 2. 2. Flagella when present is of whiplash type, and in Anthoceros the antherozoids have whiplash type of flagella. Third is a simple thalloid gametophytic plant body. These three characters are only common to algae and Anthoceros. Now, the other four characters: presence of chloroplast containing chlorophyll A and B as their photosynthetic pigments. Next, chloroplast surrounded by two membranes, starch as the primary food reserve, and lastly, cellulose-based cell wall. As I've mentioned already, I think the last four characters are continued in land plants. Characters that may be considered as forerunners to pteridophytes. Anthoceratopsida, the group, including Anthoceros, possess advanced sporophyte, which shows an abrupt leap from the sporophytes of Hepatopsida. Now, what is advancement? 1. The progressive sterilization of sporogenous tissue, which we have seen already happening in Hepatopsida, but here it is still more advanced, that is, it gives rise to differentiated vegetative structures not found earlier. This is a clear evolutionary trend. That is number one. 2. The capsule wall of Anthoceros is multi-layered with cutinized epidermis having stomata. This is also first in Bryophyta. Number 3. Internal to epidermis are parenchymatous cells having chloroplasts with intercellular spaces similar to land plants. The above two characters equip them to be partially independent of the gametophyte. Now the fourth character, the sporophyte, shows indeterminate growth due to a basal meristematic zone which seemed to be a forerunner of cambium of land plants. Next we come to the fifth point, that is the presence of columella. This we know which is a system for mechanical support and also, more importantly, conduction of water and nutrients from the basal gametophyte to the sporophyte, and therefore it seems to be a forerunner of vascular system of land plants. Now, what does the vascular system of land plants do? For example, xylem. Xylem conducts water and nutrients from the soil. So it is obvious that this columella is the forerunner of vascular system of higher land plants. Next is the symbiotic association of nostoc, a cyanobacterium, nitrogen-fixing cyanobacterium, which provides nitrogen to the Anthoceros. And what does Anthoceros provide? It provides carbohydrates to nostoc. This is therefore a symbiotic association, and it represents an early evolutionary adaptation. Next is the fossil evidence. Anthoceros shows resemblance to the Devonian fossil of pteridophyte Rhynia, because the latter plants are rootless, leafless, simple and erect structures. Lastly, the genetic evidences. The information obtained from sequenced genome of Anthoceros eliminates early land plant evolution. This we will be discussing in further detail in the next page. Now we come to the genetic insight. The genetic data from hornworts or Anthoceratopsida helps to eliminate the gradual development of crucial traits, that is, crucial characters, features necessary for survival on land, enabling transition from aquatic to terrestrial environment. Now, phylogenomic analysis place hornworts or Anthoceratopsida as a sister clade to the lineage containing liverworts and mosses, that is Hepatocopsida and Bryopsida, suggesting that they represent an ancient and early diverging lineage among the land plants. That is, they are moving apart from a common ancestor, is growing parallelly, not converging or not continuing from one to the other. That is, they represent an ancient and early diverging lineage among the land plants. Now we come to the next point, that is the closeness to algae. Now we have already discussed about the closeness to algae, that is about pyrenoids, chloroplasts and so on. Now something about the genes. Now chloroplast genome, while the gene content in organization of Anthoceros chloroplast is similar to the land plants, the specific RNA editing patterns and an unusual introns are more closely related to some green algae. So what does it say? That gene content and the general organization, genetic organization of chloroplast genome of Anthoceros are similar to land plants, but the specific RNA editing patterns and one unusual intron are closely related to some green algae. Now we must be remembering RNA editing. What is RNA editing? First of all, you must be remembering that we get RNA from DNA by the process of transcription. Now after the transcription, that is post-transcription, the RNA gets edited. For what? For a number of reasons. That is the introns. What are the introns? Introns are non-coding regions. These are removed, and exons, that is the coding ones, are spliced together to form the mature RNA. So these RNA editing patterns, some specific RNA editing patterns and an unusual intron are more closely related to some green algae. So what does it say? It says the gene content and genetic organization of Anthoceros chloroplast similar to land plants, but the specific RNA editing patterns and an unusual intron are more closely related to some green algae. Now next is the cyanobacterial symbiosis gene, LCIB. This have already been discussed, but it is also discussed here because it is about genes. So you can take that portion from there and insert it here. Now I repeat here, genetic studies have found that Anthoceros and algae, green algae, share a key CCM, that is carbon concentrated mechanism gene, LCIB, in their pyrenoids, which has been lost in other land plant lineages and later replaced by more efficient systems in higher plants. Now this we will be discussing later, and we'll be explaining this CCM later in the last page. Now we'll come to the next point, unique genome structure. Hornwort or Anthocerotopsida genomes exhibit unique features. What is that? Such as the lack of repeat dense centromeres that is characteristic of vascular plants. So all vascular plants have in their genome repeat dense centromeres. That is, the genetic structure of the centromeres have repeated sequences, and that is necessary for their structure and function. That specific type of genetic assembly, that is the repeat dense centromeres, is absent in hornworts. So this is unique for Anthocerotopsida genome and is not found in land plants. Land plants we have repeat dense centromeres. Next point, evidence for genetic homologs and unique traits. Genetic homologs is same genetic ancestors. Now the following are the evidences for genetic homologs and unique traits. First, genes for later plant adaptations. Anthocerotopsida or hornwort genomes contain conserved genes related to meristem and stomatal function, also found in later land plant lineages. This suggests shared genetic mechanisms for these structures. So this has also been discussed earlier, except we haven't mentioned about the genes. Now conserved genes, what is conserved genes? Conserved genes, those genes that remain unchanged over the evolutionary period, and that is because they have a very critical biological function, and critical biological function is present in all organisms, so that they are conserved, not changed. So I repeat, Anthocerotopsida or hornwort genomes contain conserved genes related to meristem and stomatal function, also found in later land plants lineages. This suggests shared genetic mechanisms for these structures. Now the next point about the common ancestor genetic evidence of common ancestor is the symbiosis genes. The genes for arbuscular mycorrhizal symbiosis was present in the common ancestor of land plants and is shared between Anthoceros and angiosperms. This supports the idea that beneficial relationships with fungi were necessary for colonizing land. Now, what is arbuscular mycorrhizal symbiosis? This is a mutualistic relationship between most terrestrial plants and soil-borne fungi where they exchange nutrients and therefore both are benefited and they help growth of plants and as well as the fungi. Next point is the horizontal gene transfer. Horizontal gene transfer is the transfer of genes not from parents but from other organisms. The Anthoceros genome contains signatures of horizontally transferred genes from bacteria and fungi, such as those related to stress response like drought tolerance and defense. This therefore suggests that acquiring foreign genes was an important evolutionary mechanism for early land plants to adapt to terrestrial habitat. Now, this horizontal gene transfer enable one to evolve fast by acquiring a trait from other organism and for adapting to the environmental conditions. Now, the combination of ancient and more derived genetic traits in Anthoceratopsida or hornworts provide critical insights into the genetic toolkit and adaptations that were present in the earliest land plants. Now, the glossary, and this is particularly for students who have come from different streams. That is, this is the explanation of some terms and that is not to be included in the answer. I have already explained to some extent, but here this is done in a more clear way. First is the carbon concentrating mechanism or CCM. CCM uses active transport systems, that is transport involving ATP, to move inorganic carbon, both CO2 and bicarbonate, in the anionic form from the environment into the cell, where carbon anhydrase, an enzyme in carboxysomes. Carboxysome is a protein compartment found in cyanobacteria and other bacteria. To convert the more abundant bicarbonate into carbon dioxide or CO2. CO2 is then transported to the site of photosynthesis to create a localized high concentration of CO2. High CO2 concentration level ensures rubisco's preferential binding to CO2 over O2 and leads to efficient carbon fixation, the first step of photosynthesis, and thereby reducing photorespiration. Now, rubisco is an enzyme which the full form is ribulose 1,5-biphosphate carboxylase oxygenase. That is, if it binds to carbon dioxide and converts ribulose 1,5-biphosphate to glucose, then it is doing the carboxylation function. But if it binds to oxygen, that is the oxygenation function. So, ribulose 1,5-biphosphate carboxylase oxygenase is the name of the enzyme. So, in high CO2 level, it ensures rubisco's preferential binding to CO2 over O2 and leads to efficient carbon fixation, the first step of photosynthesis, and thereby reducing photorespiration. So if the rubisco binds to CO2, what happens? ribulose 1,5-biphosphate is converted to hexose sugar, that is glucose. That is the first product of photosynthesis, and thereby photorespiration is stopped. This enables survival in low CO2 environments. So this is all about, for now, carbon concentrating mechanism, or CCM. Then we come to the term genetic homologues. What are genetic homologues? These are genes, or other words, genetic sequences that originated from a common ancestral gene. Next is the arbuscular mycorrhizal symbiosis. Now this is a mutualistic relationship, symbiosis, between most terrestrial or land plants and arbuscular mycorrhizal fungi. Now what are arbuscular mycorrhizal fungi? Fungi which enters the root of the plants, that is mycorrhiza. Myco is fungus and rhiza is root. And then it forms a structure known as arbuscles. So, we read again, arbuscular mycorrhizal symbiosis is a mutualistic relationship between most terrestrial plants and arbuscular mycorrhizal fungi. Now, what is this mutualistic relationship? These soil-borne fungi enter the plant's root cells and receive carbon from the plant, while in return it helps the plants absorb water and essential nutrients, particularly phosphorus. This ancient association is vital for plant growth, for ecosystem sustainability, and plants' tolerance to abiotic drought, abiotic stress like drought and salinity. So these characters are fast equipped to for migration to land and to tolerate abiotic stress like drought and salinity. The next come to horizontal transfer of genes. Horizontal gene transfer is the movement of genetic material between organisms that are not offsprings. That is not parent to child, but from one organism to another organism not related. This process is common in bacteria and also occurring in eukaryotes. Unlike vertical gene transfer, that is from parent to child, horizontal transfer of genes involves acquiring new DNA from other organisms, contributing significantly to evolution by allowing rapid acquisition of new traits. Now, clade is a grouping that includes a common ancestor and all the descendants living and extinct of that ancestor. This clade is a group of organisms which have a common ancestor, and these may include living or extinct members. The next is the photorespiration, a light-dependent metabolic pathway in plants and other photosynthetic organisms, where the enzyme rubisco ribulose biphosphate carboxylase oxygen binds oxygen instead of carbon dioxide, consuming energy and releasing carbon dioxide while processing the resulting toxic byproduct 2-PG, that is 2-phosphoglycolate. So here what happens, instead of fixing CO2 in presence of light, it binds to oxygen, thereby photosynthetic process is hampered. Next is a repeat dense centromere. This is a chromosomal region or domain that is characterized by a high concentration of repetitive DNA sequences, such as satellite repeats. What is a satellite repeat? This consists of large arrays of tandemly repeating non-coding sequences. Tandemly repeating means that is repeated sequentially. Small base pairs repeated sequentially. This is satellite repeats. But these repeats represent non-coding regions. So this is satellite repeats or long terminal repeat LTR retrotransposons. Now transposons, as you know, are mobile genetic elements. Now what is retrotransposons? Retrotransposons are mobile genetic elements that copy themselves from RNA via reverse transcriptase and insert the new DNA copy into the host eukaryotic genome. A long tandem repeat, that is, it is long and it is long terminal repeat, that is, it is positioned in the terminal area. And so these repeat rich areas are fundamental to centromeric structure and function. Now I think it is in this context it is more useful to know this as a repeat dense centromere is a chromosomal region, a domain that is characterized by a high concentration of repeated DNA sequence. That's all. And these repeat-rich areas are fundamental to centromeric structure and function of eukaryotes. That is, we are talking of eukaryotic centromere structure. Now, this type of structure is lacking in Anthoceros. So, Anthoceros lacks this evolved character. Lastly, we come to conserved gene. A conserved sequence is defined as a segment of DNA or protein that has remained largely unchanged throughout evolution, indicating its critical biological function. Because critical biological functions, just fundamental functions, don't change. So those genes also don't change and they are conserved to its original form. Now I want to say a few words before I just end my talk. That is, if you think that this presentation is useful to you, please like and subscribe this channel. And also, please comment in the comment section that what topics you need more, whether it is gametophytic evolution in bryophyte, or it is some topic of microbiology, that is prion or viroid or anything like that, or some other. So that is the end. Thank you.